Hi, I apologize for not being there in person. I had some flight issues, but today I'm going to talk about our work on using our iPSC model for Jansen de Vries Syndrome. And iPS cells, in case you don't know this already, are cells that resemble embryonic stem cells in their capacity to differentiate into any cell type in the body. And embryonic stem cells are considered for use in regenerative medicine to grow cells and tissue to replace those damaged by disease or injury. But embryonic stem cells are controversial because they are derived from a 1-week-old embryo. So an alternative was invented by Dr. Shinya Yamanaka in 2007. He was able to find a method for converting normal cells in the body into iPS cells using these different genes introduced into those cells. This was created quite a great deal of buzz. This is the paper that was published in 2007 describing those results. And it was such an important discovery that it led to a Nobel Prize only 5 years later, which he shared with John Gurdon, who had done some basic studies in the 1950s that led the way to Shinya's discovery. These studies by Gurdon were carried out on, believe it or not, the frog eggs. So that shows you the connection between different biological systems and neurodevelopmental disorders. Even though this was conceived as a method for producing cells for regenerative medicine purposes, people like me interested in autism and psychiatric disorders and neurodevelopmental disorders looked at this as a great opportunity to make patient-specific neurons in the lab. So basically, in our lab and in many labs throughout the world, we are growing schizophrenia neurons, and we're growing autism neurons, and I'm growing JDVS neurons. This is a monumental leap in our ability to study these conditions. So we have developed iPS cells from blood cells, isolating this fraction of cells called CD34 positive cells, which are the same cells that are used in bone marrow transplants. We use this virus containing the Yamanaka reprogramming factors to make these iPS cells. And we've generated lines for patients and controls. And we've also used CRISPR technology to introduce the Jansen de Vries mutation into control cells, which has major advantages for a variety of reasons I don't have time to go into. CRISPR, in case you don't know, is a gene editing tool that allows investigators to literally introduce a mutation into any gene at any site in any cell. It's a very, very powerful technique. We use that to generate a Jansen de Vries mutation in exon 5. And here's a protein gel showing 5 samples. These are control samples. This is the native PPO1D protein, and this is the truncated protein produced by exon 5 mutations. Here's one patient line, and here are 2 CRISPR lines. We've also generated a line from a patient who was a mosaic for an exon 6 JDVS mutation, and we were able to isolate cells that contain 2 copies of the normal PPO1D gene and cells that have 1 copy of the exon 6 mutation. This is a very, very important cell line that I will share to other investigators, and I don't have time to go into why this is such an important cell line. And CRISPR was also a major discovery, and it led to a Nobel Prize in 2020 for 2 of the major players, Jennifer Doudna and Emmanuel Charpentier. You should know that the CRISPR system was derived from bacteria. Many bacterial species have a CRISPR system for destroying the DNA of incoming viruses, which infect bacteria as well as humans. and the specific type of virus that infects bacterial cells or bacteriophage. In any event, it's such an amazing story to go from this immune system in bacteria to editing the human genome. So our basic strategy is to take these iPS cells and differentiate them into two cell types that we feel are important, and I'll discuss those in the next slide: glutamatergic neurons and microglia. And we use a technique called proteomics and a similar technique called phosphoproteomics to look for proteins that are differentially expressed in these cells in patients versus controls. And the main purpose for this is to understand the underlying molecular basis of JDVS. In other words, what is the truncating protein, the truncating PPOND protein doing downstream, and maybe even identify novel druggable pathways and proteins that might be targets for therapeutic intervention. And the proteomics and phosphoproteomics studies use a mass spec, which is the ability to analyze thousands of proteins at the same time. So it's a very powerful tool for studying the molecular basis of disease. And phosphoproteomics is an analysis of phosphorylated proteins. Now, many proteins are living inside of cells in an inactive state, and adding a chemical group called phosphate can activate those proteins. And it's a major homeostatic process in cells. PPOND is a phosphatase that removes phosphate groups from a certain group of proteins. And this balance between phosphorylation and dephosphorylation is important in the life of a cell. And when it goes in the wrong direction, as is the case with these truncating mutations, which have an increase in phosphatase activity because of an increased half-life, then you have problems. So here are the two cell types that we are focusing on. These are glutamatergic neurons. The green dye is just a stain for a marker for these neurons. This is the major excitatory neuron in the brain, and it plays a really important role in behavior, cognitive function, learning, and memory. And mutations in the genes that affect these neurons are very common in autism and other NDDs, neurodevelopmental disorders. Microglia are the immune cells of the brain, and they protect it from infections. But they actually play a much more important role in brain function, since infections in the brain are not very common. They are involved in a process called synaptic pruning, which is the destruction of unused or underused synapses. They also are involved in removing cellular debris from cells and from the outside of neurons that can lead to neurodegenerative disorders if they accumulate, like the beta amyloid protein in Alzheimer's disease, AD, and alpha-synuclein in Parkinson's disease, or PD. And alterations in synaptic pruning are found in many NDDs. And this is a process that goes on in the developing brain. There's almost as much destruction or pruning of neurons as there is development. And in the post-development brain, it's involved in, again, learning and memory and other cognitive functions. Another reason for looking at microglia is that we have found that a small fraction of JDVS patients have a severe acute behavioral decompensation that looks like a neuroinflammatory disorder. This is very rare. We think that other mutations, in addition to the Jansen de Vries mutation, have to occur in order for this to happen. But that's something that my lab is interested in. And this is a very rough cartoon sketch of how microglia interact with these dendritic spines. These are the portion of the neuron that grow and change during learning and memory, and they basically eat these underused synapses in the way that the white blood cells will eat bacteria in the periphery. And if you remove or incapacitate the microglia in mouse models, you get a severe defect in learning in those animals. And another reason for looking at the microglia is that they resemble immune cells found in the blood and various tissues called macrophages and monocytes. They, in fact, microglia are derived from cells that give rise to these cell types. And these cells in the theory kill bacteria by engulfing them by phagocytosis. And we thought that maybe we can use microglia as a proxy for understanding why some kids with JDVS have recurrent infections and why PPMD mouse models showed major defects in immune function. So we did proteomics analysis on control and the JDVS neurons and microglia. And for the microglia, we also analyzed those cells after stimulation with a drug called LPS, which mimics a bacterial infection and mimics non-infectious triggers of the immune system. And we identified roughly 5,000 proteins or so with mass spec. We see, I hope you can see my cursor, we see most of the proteins don't show any differences between patients and controls, but the colored circles here represent proteins that were differentially expressed, either increased in the PPO&D samples or increased in the controls. And they range from several dozen to several hundred, depending on the analysis. And when you're looking at many proteins and genes and RNA molecules through these techniques, either mass spec or other technologies, it's very important to look to use these tools, these bioinformatics tools, to understand patterns of gene expression, patterns of protein expression that you can't really see, the human brain really can't visualize. Two of the tools that we use were KEGG and the gene ontology, or GO, and these identified patterns of differentially expressed proteins and differentially expressed phosphoproteins. And these analyses are based on tens of thousands of different published studies. But I also find it very useful to evaluate individual proteins because we can identify certain patterns that the computer programs cannot, as you'll see in a few minutes. So this is the result of the GO analysis of phosphoproteins in neurons. And I'm only showing you a small fraction of the data because it can be quite overwhelming. And the two major findings here were that there were proteins that were involved in a process called RNA splicing. Splicing is a process of converting RNAs into those that can be translated into proteins. I won't go into that in any great detail, but 90% of genes are regulated by some kind of splicing mechanism. And there are certain groups of NDDs that are problematic because of mutations in neuron-specific splicing factors. And many tumors have splicing abnormalities. But the main finding here, the main significant finding, was that there were proteins that are involved in cytoskeletal organization. The cytoskeleton is exactly what it sounds like. It's the skeleton of a cell. Instead of having a bone structure like the skeleton of an animal, the cytoskeleton is made up of proteins like actin and tubulin and other proteins that give the cell its form, its body. And as a cell grows, as a neuron grows, the cytoskeleton has to be functional. And as dendritic spines form, the part of the neuron involved in learning memory, one needs to have an intact cytoskeleton structure. And the finding that there are many proteins that disrupt cytoskeletal organization suggests that these processes are problematic in JDBS. So we validated this by doing something called a neurite outgrowth assay. And without going into detail, this showed that there's a decrease in the length and branching of neurons formed from our iPS cells. We have not been able to analyze dendritic spines because these neurons are rather immature, and the spines that are formed without technique are kind of wimpy. So we need to find a better way to visualize the spines. And here is a cartoon of a dendritic spine. It looks complicated, but actually this is actually a simplified version of a spine. This is the postsynaptic spine, which receives transmitter from a presynaptic neuron. And all the action takes place over here. Learning and memory, various behavioral functions all occur at this dendritic spine. And these longitudinal structures are the proteins that form the cytoskeleton. This is, you can see, this is the PSD, which is an organized structure made up of dozens and dozens of proteins. And one of the more common types of mutations found in autism and in other NDDs are mutations that affect the PSD. For example, SHANK3, one of the major autism genes. So this is a major player in these conditions. The KEGG analysis also showed splicing to be a major problem. The spliceosome is one aspect of splicing, but it also identified some signaling pathways that might be targets for therapeutic intervention, like the ERB pathway, which I'll show you in a minute. But more importantly, some of the proteins that would differentially expressed in neurons also have been implicated in these different neuro developmental disorders. It doesn't mean that kids with JDDS or young adults with JDDS are going to have this problem. It just means that the pathways for JDDS and Parkinson's disease, for example, overlap. And I'll go into that in more detail a little bit later. And this just shows cartoons of ERB signaling and neurotrophin signaling, which are neuron-specific growth factors. Again, the downstream targets of these, which are hard to see on the slide, but they may represent targets for therapeutic intervention. This is the GO analysis for the microglia phosphoproteomics. Again, splicing is coming up as the major player. So this is a very robust finding in different cell types. But the one that was most intriguing to me was this collection of proteins that are involved in the DNA damage response. And as you may know, PPOND is a major player in that response through its regulation of p53. And these other proteins, CHEK1, CHEK2, ATR, and ATM, are also involved in regulating the DNA damage response with DDR. And these are connected directly or indirectly to PPOND. And the reason why I found this to be quite intriguing was that we have identified several PANS patients who have loss of function mutations in CHEK2, ATR, and ATM. And in the case of ATR and ATM, it's 2 or 3 individuals. We only found one with a CHEK2 mutation. And this shows you the connections between PPOND, P53, ATM, ATR, and CHEK1 and CHEK2. Now, DDR is a normal physiological process in cells. As cells divide, as cells get exposed to various types of stress like UV radiation or oxidative stress, DNA damage occurs. And that damage is repaired by the DNA damage response pathway, of which PPOND and P53 are major players. There's also a process involving DNA present. This all happens in the nucleus of the cell. In the cytoplasm of the cell, in immune cells, and in microglia, there's a DNA sensing mechanism where that recognizes viral DNA that invades cells, and that triggers an immune response. Typically, the antiviral cytokines, type 1 interferons, are activated by this process. This process is connected to DDR, and that when you have impaired DDR through mutations, let's say in ATM and P53 and possibly PPL1D, this can lead to the leakage of DNA from the nucleus into the cytoplasm, and that can trigger this DNA sensing pathway and lead to an autoimmune or autoinflammatory disorder. And lupus is an example of that. There are subgroups of lupus that have mutations in genes that affect the DDR. And we think that a similar process might be going on in kids with PANS and in the subgroup of patients with JDDS who have an acute neuropsychiatric breakdown. This these are the results of the proteomics and phosphoproteomics analysis of LPS stimulated microglia, the drug that mimics a viral infection. And again, we see splicing and the DDR as major players in this pathway, in these cells. And we also found, somewhat surprisingly, that there's negative regulation of interleukin-6 or IL-6 production. IL-6 is a major pro-inflammatory cytokine that's been implicated in neuroinflammation. So since we're looking at a neuroinflammatory process in a subgroup of patients with PANS, with the JDDS, we were expecting to see an increase and not a decrease in IL-6. And this is a cytokine assay we did in the microglia. Actually, it was the cytokine released by microglia in response to LPS. And you can see, I explained to you that there's an increase in the expression of several of these cytokines in control cells. These are control cells, baseline control cells with LPS, control baseline, and then LPS. A big increase in these cytokines and similar proteins called chemokines. But in the JDDS samples, we don't see as much of an increase. And here's IL-6. Again, I hope you can see my cursor. IL-6 is in the middle of this slide, and you can see that there's an increase in IL-6 in the control samples, but no increase at all in the JDDS samples. So rather than seeing this marked increase in these cytokines and chemokines, we see actually a blunted response, and that's not that troubling to me because we know that in the brain and in the immune system, too much or too little both can lead to problems. Here's the KEGG analysis in microglia, again showing a connection to these proteins that are differentially expressed in the Jansen de Vries microglia with neurodegenerative disorders. Again, ERP signaling is coming up, and connections to different infections, including flu and COVID, also were found in these cells. And we also see proteins that are involved in oxidative phosphorylation, and that suggests the possibility of some mitochondrial dysfunction that maybe Dr. Bowles will talk about today. So here are some of the proteins that were differentially expressed in the microglia, and it's a rather complicated slide. I picked out the top 15 or 20 proteins. I'm only going to mention the most important ones. We found that proteins involved in the regulation of ubiquitin were the primary proteins that were differentially expressed. So these three ubiquitin proteins, ubiquitin regulators, especially UBR4, were abnormally expressed. These are ubiquitin, E3 ubiquitin ligases. And these ligases attach this protein called ubiquitin to proteins. And that leads to a variety of regulatory pathways, but the ones that are most important for this session are the DNA damage response and proteasomal degradation. So proteins get damaged from oxidative stress and other stressors. Those proteins get tagged with ubiquitin, and that ubiquitin tagged molecule gets destroyed and recycled by an organelle in the cell called the proteasome. And if this process is impaired, these abnormal proteins accumulate in cells, and that is could cause problems. And these other proteins were also the major differentially expressed proteins. Some of them are associated with autism, and NUX1 is associated with Parkinson's disease, as well as DNA repair. So these are the proteins that led to those KEGG and GO findings. And the two proteins that were most dysregulated in microglia, the previous slide showed neurons, were a gene called POGZ and again UBR4. So UBR4 is differentially expressed in both neurons and microglia. POGZ, loss of function mutations, are a well-known cause of autism and other neurodevelopmental disorders. And most intriguingly, Dr. Bowles has identified this as a possible candidate gene for cyclic vomiting syndrome. So that connection to me is not coincidental. Now, POGZ forms a complex with another protein called ADNP, which regulates the expression of genes involved in glutamatergic differentiation. If you knock out these genes in mice, you get a neuroinflammatory process and dysfunction of microglia and a decrease in glutamatergic transmission. And since we found these proteins to be differentially expressed only in microglia and not neurons, we suspect that this decrease in neurons in microglia rather is playing a role in the various cognitive and psychiatric manifestations of JDDS. Now, UBR4 is a major player in proteasomal degradation proteins, as I alluded to in a couple of slides ago. But here's a really fascinating connection. UBR4 is involved in the proteasomal degradation of a protein called PINK1. PINK1 is a regulator of mitophagy, which is the destruction of abnormally functioning mitochondria, a normal physiological process. And it does this with this protein called parkin. And mutations in PINK1 and parkin are found in rare forms of hereditary Parkinson's disease. You have two copies of loss of function mutations in parkin. You have early onset Parkinson's disease. We have two patients, I've identified two patients with one copy of a parkin mutation in PANS. Again, another connection between PPOND, neuroinflammation, and ubiquitin. So the UBR4 findings suggest that mitochondrial dysfunction is playing a role in Jansen de Vries syndrome. So in summary, the proteomics analysis identified POGZ and UBR4 and a few other proteins as candidates for the neurodevelopmental and psychiatric manifestations of JDDS and alterations in the cytoskeleton, which may translate into dendritic spine abnormalities, is a major player, it seems. And we've identified novel primary and secondary targets of PPOND, including the DDR, which is known already, but splicing and mitophagy, which are not that well recognized. And we think that some of the same changes that we find in microglia, if they're occurring in monocytes, might account for the increased risk of infections seen in some patients with JDVS. And we also identified proteins that overlap between JDVS on a molecular level with neurodegenerative disorders. And in terms of future directions, we are now testing PPO-ND inhibitors to see whether or not some of these abnormalities that we found in neurons and microglia can be reversed. We also are considering ERB and neurotrophin regulators as potential translational targets. But a lot more work obviously needs to be done. All of our work has been carried out on a shoestring budget. And we need mouse models, more microglia studies, and we also need to analyze the dendritic spines in these neurons and carry out electrophysiological studies. And that's going to require more NIH grants. And we think that the connections that we found between a rare disorder, JDVS, with a more common process like neuroinflammation and Parkinson's disease might increase the odds of a successful grant application. And actually, I already have one small NIH grant based on our findings. I'm trying to get money for a mouse model. And the other thing that's important is that all of these genes of interest, PPO-ND, POGZ, ADMP, and UBL4, are primarily expressed in the cerebellum. The highest level of expression is in the cerebellum. So we need to study the cerebellum as well as cortical neurons to understand how PPO-ND mutations influence behavior. And I want to acknowledge the families who contributed to the iPSC research and startup funds from the Ryan Stern Foundation, and my crew, especially Erica Pedrosa, who does most of the work that I showed you. And I again apologize for not being there in person, and I'll be glad to entertain questions at a Q&A session later or by a separate Zoom conference.